All right, guys, go ahead and find your seats. Let's finish this guy up. Everybody feel better after home heat? Yeah, no. Get some coffee. You guys are still tired. Is there coffee in there? Like, you need to go make some? Stealth viruses continuing. They can be potential bioweapons by 2035. If you are into the UN documents, 2030 is when a lot of the big changes are supposed to happen. So 2035 is a couple years add on to a lot of the, like the big global tech goals. And it's really not that far away, right? It's almost 10 years or two now. That's not that far away. We have some of this technology today on a wide scale by 2035. There's still a lot to be learned about the triggering mechanisms. Like you said, there's the potential for the hot, cold extremes to be the triggers. But again, they've got a few years before they're ready to be implemented. But imagine a genotype or a phenotype type weapon that gets released into, say we're at war with China, like the next 10 years. We release something over there that only targets them, but we have our American troops on the ground. Not to worry about it too much, wouldn't it? Scary thought. So this next one, we're moving to the injectable mesh. So think of Neuralink. Elon Musk, his little thing. He actually just the other day said that he's looking for human guinea pigs to give it a try. They have done successful Neuralink with, I think it was a chimp, and the chimp played Pong while they were staring at the TV screen. Kind of cool, right? So, like, the transition from just pure human to like a cyborg hybrid is pretty close to where we're at. So this one is super boring, but I'm just going to kind of skip through it real quick.
This method can help answer key questions in the field of neuroscience, such as how aging processes manifest in the brain, how the brain develops, and how brain diseases originate and progress. The main advantage of this technique is that mesh electronics do not elicit a chronic immune response in gliosis once implanted into the brain, and allows for seamless integration with the surrounding neural tissue. Demonstrating the procedure will be Tao Zhou, a graduate student in the Libre group, and Jungmin Lee, a postdoc in the Libre group.
What's up? What's up?
To begin the procedure, load a clean silicon wafer into a thermal evaporator and evaporate.
So basically this whole first part of the video is telling you how the mesh is made out of this disc.
2000.5 negative photo.
We're just gonna kind of jump through it a little bit. Just know that they dump a lot of stuff on the disc and then they wash it off. You can kind of see the lines there. That's the mesh.
Repeat spin coating lithography. Then, transfer the mesh electronics to a fresh beaker of deionized water at least 3 times to ensure rinsing.
So you can kind of see that goldish thing there in the water, right?
To load the mesh electronics,
that was the mesh that they're going to inject into the mouse.
Capillary needle into the 100 milliliter beaker of PBS containing the mesh electronics. Position the end of the needle near the input-output pads of a mesh electronics probe and manually retract the syringe to draw a mesh electronics probe into the needle. Push and pull the syringe plunger while it is still immersed in saline to adjust the position of mesh electronics within the Mix a clamping substrate to the skull with dental cement. An approximately 1 millimeter wide cut in the substrate improves the reliability of the folding step.
That's the mouse.
In the procedure. Afterward, mount the pipette holder with the needle containing mesh electronics into the stereotaxic frame using a right-angle end clamp. Attach the side out.
A lot of this is for
Then,
like his
stop the
doctorate or
flow
whatever
from
it
the syringe
is.
pump. In this procedure, use the stereotaxic frame to carefully guide the needle to the flat, flexible cable clamping substrate and across the gap, flowing the solution with the syringe pump to generate a slack in the mesh electronics interconnects. Once the needle is above the clamping substrate and across the gap, resume the flow substrate that will hinder the insertion into the PCB-mounted 32-channel ZIF connector. For freely moving recordings, release the mouse from the restrainer after inserting the preamplifier PCB and grounding the reference screw. Record for the desired length of time using the data acquisition system while the mouse behaves freely.
I know, poor little mouse, right? Had that big old thing on his head.
Shown here are the representative local field
It's what?
potential heat maps from
It's
the
his PC, yeah.
32-channel mesh electronics probes
No laptops
injected
for it
into the
yet.
mouse hippocampus and somatosensory cortex. Data were recorded while the mouse freely explored its cage at 2 months and 4 months post-injection. Local field potential amplitude is color-coded according to the color bar at the...
While attempting this procedure, it's important to remember to test the I/O interfacing to confirm successful electrical connection to the mesh electronics probe. The measured impedance helps troubleshoot the source of any problems and is critical to identify issues with interfacing or fabrication. Following this procedure, histology can be performed in order to study the cellular environment around each recording electrode. Unlike conventional rigid brain probes, mesh electronics can be left in the tissue during histology, making it possible to precisely correlate electrophysiology data with immunohistochemical analysis.
A lot of big words, right? Basically, what this is getting at is in the future, we're going to be able to plug into a computer and see deeper into your skull.
So the mesh is going to be available to all of us eventually down the road. Do you know what Neuralink is? Elon Musk. Okay, so he owns a separate company called Neuralink. What they're trying to do is hook up the human brain directly to a computer.
Also, mesh is like the...
Yeah, so this is like one of the very basic steps in trying to do it. So instead of having to type on the computer, you just sit there and think about what you want, and whatever you want to have on the screen. Okay. These last two documents that I used to make this are super technical and like a lot above us, but it's a thing that's out there. That's again the big thing to why I made this. Things are coming down the pipeline that we may not understand, but like our kids are going to know exactly what's going on. It's like, how do I put the clock on the VCR correctly? I hate this. You ever VCR? Kind of one. I put that one out there for you old guys.
There's those on
I don't think she knows what it is. She's 5. No, so like one of the advantages of what they were talking about in the document was, in the past, somebody had to create these MRIs, fMRIs, right? Those all map the brain. What the mesh they're trying to work on is digging a little deeper inside the brain, looking more at a smaller scale, trying to figure out little things. So imagine if you had Parkinson's or Alzheimer's or something like that. Inject that mesh in there, be able to figure out what's going on real quick, right? So this document, it was the most technical out of all the ones I read. Basically, just be aware that in order to get the things that we want to get, the mesh has to happen in order for like the nanosensors and Tony Stark type nanobots. Could be Doctor Strange. Put a little magic on there, never heard, right? So this is what it looks like. So you have your nanowires, then it eventually goes to a scaffold, and then it becomes fused with tissue. So look like the mouse video, for example, they just put the whole mesh in there. So you wouldn't have necessarily a Tony Stark where it just all like comes out of this little thing. You would just inject this scaffold, and then it becomes combined with your tissues. DARPA, if anybody knows what that is, has created an implantable gel that can detect the COVID in your body already. They also have the technology to hook you up to a dialysis machine that's FDA approved apparently. That'll clean out all the blood of all the COVID stuff. Why that's not on a wide scale, I don't know. Colonel Matthew Hepburn, he gave an interview to 60 Minutes. That's where I learned about that. That little green thing right there is what the DARPA has created. But in the future, yes, this is Buzz Lightyear. I figured Buzz Lightyear is more appropriate than Master Chief. But imagine you as the medical field can flip open your little scanner, scan it over Hardwick. Oh, he's going to the sepsis, he's got tuberculosis. I know how to fix this now.
You have Trek words in Star Trek.
Oh, okay, I wasn't thinking Star Trek. I'm sorry. The one time I did it.
You're a Trek word.
Does anybody have any questions? Okay. Thanks, guys.
Appreciate it. It's on its own time. It wasn't out of the way of